Thank you, Jean-Pierre. And thank you for the topic. I would like to start out by saying that the speakers on this platform are the world authorities on the molecular genetics of pediatric brain tumors. And I want to make sure that you don't confuse me with any of those people because my knowledge of the molecular genetics is limited. But as a clinician, I can give this whole talk with just this one slide because I can say that molecular biological understanding of these tumors has changed our whole world. The end. That's all we need to know. Um, but I'll go through some thoughts as a practitioner and uh, try to leave some time at the end. So first, the biological manipulation of tumors based upon their molecular genetics is not exactly new. If you look at the ability to shrink prolactinomas uh, based upon dopaminergic agonists, this goes back to 1985. So for 35 years, we've been making these tumors shrink. And to date, these are no longer surgery first tumors. These are medically treated tumors, and the majority of them will respond quite nicely to medical therapy. Likewise, it's been about 15 years that we've had an understanding of tuberous sclerosis and the SIGAs, and we know that with everolimus or other mTOR inhibitors, we can now get these tumors to shrink, and we have a number of these children in our TSC clinic that are being managed by neurologists, not even oncologists, where they're placed on medication for their tumors, and the tumors respond nicely. So they're not being referred to surgeons anymore unless they have side effects from the medication or they don't respond to the medication. So I would say that molecular genetics has completely changed our field and will continue to do so into the future. And there may come a time where the role for surgeons is much less than it is today. So uh, Dr. Mack alluded to the fact that historically we've relied upon what we see under the microscope to try to differentiate tumor types and to determine both prognosis and therapy. And we've also noted for a long time that what we see under the microscope doesn't necessarily predict how these tumors are going to behave. And I'll point out that myxopapillary ependymomas, particularly in children, do not behave like a grade 1 tumor, WHO grade 1 tumor. They are prone to metastasize, they're prone to recur. And in fact, we hear that in the next iteration of the WHO classification, this is going to be a grade 2 tumor and no longer classified as a grade 1 tumor. So this is a paper from David Ellison where they looked at the agreement from the world authorities in neuropathology as they did central review of children enrolled in our national cooperative studies and demonstrated that even amongst the experts, there's a 20% variable in their interpretation of what they see looking under the microscope. And this is a slide that Mike gave me years ago that I've used quite often, showing that even ependymomas in different areas of the brain, posterior fossa, supratentorial, spinal, that may have different biologies, different prognoses, look the same under the microscope. So what we've done historically has just not been that great in our management. And Dr. Mack pointed out that we now talk about 9 different subtypes of ependymomas based upon their molecular genetics. And this is a paper from David Ellison now suggesting that there are 9 different subtypes just of posterior fossa A tumors. One of those subtypes that Nada mentioned is an H3K27 variant, which is a very interesting tumor. We've had 3 of those. They're typically in teenagers, typically in the fourth ventricle, typically invade the floor of the fourth ventricle just above the obex. And personally, I don't know what the significance is. The kids that we've had with that mutation seem to be doing well with resection and focal radiation, unlike the other midline H3K27 gliomas we've had. So we're at a point now where we can take a piece of a child's tumor and send it to Foundation One and get back a report that gives us the genomic alterations on that particular child's tumor or adult's tumor. And with that report, they can tell us what the molecular abnormalities are. They can tell us what treatments are available for that, give us an explanation of what that genetic aberration is, what its significance is, what the treatments available may do, their mechanism of action, and tell us where different studies are available around the country, and how to get in touch with those investigators if we're interested in enrolling our children in these phase 1 or phase 2 studies. So our whole world has changed based on this. And I'd like to point out this paper by our moderator, Mike Taylor, that I think is a neurosurgical classic. Hopefully, every neurosurgeon has read this paper, knows this paper by the time they finish their training, but it describes the molecular variants of what for decades we thought was a solitary tumor. And yet we all knew that some children responded very well to therapy and other children, despite gross total resection and therapy, went on to metastasize and die, and we didn't understand why. But we now understand that there are molecular variants of medulloblastoma, and we have lumpers and splitters. If you look at Scott Pomeroy's lab, he says there may be 9 variants. At St. Jude, we talk about the Wnt tumors, the hedgehog tumors, and the non-Wnt, non-hedgehog tumors, so 3 variants. But by convention, we've agreed to use the 4 different variants that have different demographics and different prognosis. And over time, this has led to different treatments. This is the outcomes by different prognoses. And as you know, the Wnt tumors are almost 100% curable. The other tumors, not so much so. But in our current iteration, we've divided up the treatment schemata based upon the molecular genetics in addition to these other factors. And we had the occasion to try this smoothened inhibitor a few years ago that blocks at this level for the hedgehog medulloblastomas. And in our early experience, we had children with recurrent hedgehog tumors such as this that had dramatic responses in a short period of time to this smoothened inhibitor. Unfortunately, the duration of that response was only a couple of months, but we've now incorporated it into our treatment schema for children with those tumors. And here's an example of the clinical significance of the molecular profile. This is a child that was referred to us from Michigan who had had a gross total resection of this tumor, 5 years of age, no evidence of metastatic disease. So historically, he would have been relegated to average risk therapy. But when we looked at his tumor by FISH, we could see that he had MYC amplification, which by our current strata leads him to a high risk protocol rather than an average risk protocol. In terms of the low-grade gliomas, again, we've learned that by location, not all astrocytes that give rise to these tumors are the same. The molecular variants can be different by location, and the prognosis can be different. And when we look at our historical low-grade chemotherapy, typically carboplatin, vincristine, or whatever, we know that the chances of cure of these children is somewhere around 5% long-term. We may get responses to therapy, but the duration is not lifelong. And so we need better treatments for these children. And so as we get to understand these molecular pathways, we're coming up with monoclonal antibody du jour for treatment, and we're learning a lot about these. Solumetinib was recently approved by the FDA in the United States for use in children with neurofibromatosis type 1. But we're also learning that by dividing these children up into multiple molecular subgroups, it becomes very difficult to do multi-center randomized trials in the treatment of these children because we have too small a numbers for each group. If you divide posterior fossa ependymoma type A into 9 different subgroups, your chances of accumulating enough data on any one subgroup through multi-center trials becomes impossible. And so we're learning as we go, not only do these monoclonal antibodies have variable effects on the tumor, but they have variable side effects in the patients. And we've seen some children that start into monoclonal therapy and it affects their skeletal maturation. We had a child last week who had an ALK phos of 6,000, even though he was asymptomatic and seemed to be responding well to his monoclonal therapy. So we're learning about responses, but we're also learning that these are not necessarily benign drugs. But here's an example of an optic pathway tumor with a BRAF mutation and a tremendous response to a BRAF inhibitor, as you can see here. And so we see these, we catalog these, we don't know what their duration is. We don't know what happens when we stop the therapy, except that in many instances, the tumors can recur fairly quickly. And here's responses to entrectinib in a number of children where you can see dramatic shrinkage of tumors, both low-grade and high-grade gliomas to this monoclonal therapy. But we don't know yet what the long-term control rates are, long-term consequences of therapy are, how long to keep the kids on therapy. And we're going to learn along the way. Here is the incidence of congenital glioblastoma, babies born with these giant tumors that have historically carried a very poor prognosis. And yet, now that we have a better understanding of their molecular genetics, we note that they're not all the same, that the group 1 tumors um, may have variability in the tyrosine kinase receptor pathway and may have an intermediate prognosis. The group 2 tumors with the RAS-MAP kinase pathway involved have an excellent prognosis. And we've got others with midline glioma involvement of these pathways that have a very poor prognosis. So based on the molecular genetics of these tumors, all we can say at this point is that it's very important for us as neurosurgeons to get tissue to the lab in every instance so that these children can have a chance for us to understand what's going on with them. In that regard, I wanted to address Francesca's question about the need for biopsy, because I think in retrospect, we can say that as neurosurgeons, we created a great disservice 25 years ago when we made a decision not to biopsy these tumors anymore. We went for 25 years with no significant improvement in our understanding of these tumors or in the outcome of these children. And it wasn't until we started biopsying them again that we've come to understand what Nada's lecture was all about. And so I will make the point that with our current technology, our risk of complication from biopsies is less. Historically, we've targeted the most enhancing or adjacent to necrotic areas tumor. Well, that's also the area of the tumor that is most likely to bleed, most likely to hemorrhage and cause catastrophic complications. But historically, we were relying on cellularity as part of the WHO classification, and we wanted those most cellular areas. Now that we understand, as Nada says, that every tumor cell in that brainstem has the clonal molecular abnormality, we no longer have to target that enhancing high risk area of the tumor. All we need to do is get a piece of the tissue from a relatively safe area within the tumor, and we'll understand the molecular profile of that tumor. I'll also say that not all of these are H3K27Ms. We're seeing children with ETMRs. We're seeing children with PNETs of the brainstem. We're seeing the occasional child that has a low-grade glioma that goes on to carboplatin vincristine instead of a high-grade chemotherapy regimen for these tumors. So I think we can be safer when we biopsy these tumors, and the need to do so becomes more important nowadays. And I'll skip over this in the interest of time. And then finally, I wanted to just bring up the molecular profiling of craniopharyngiomas, because this has also changed our world, not so much in pediatric neurosurgery where we have the adamantinomatous craniopharyngiomas, but in the adult world where they have the papillary craniopharyngiomas. And we are just now coming to understand molecular targets for these different craniopharyngiomas, but we also have isolated reports of dramatic and rapid responses of adult craniopharyngiomas to BRAF inhibitors. As you can see in these slides, we only pray that we have a day soon where we have the similar target for the adamantinomatous craniopharyngiomas in the children. So it's interesting times, a lot of change happening, and I'll stop there, but would like to thank you for including me with these brilliant speakers that you have in this session.
So thank you, Rick, for this fantastic talk, giving us really the impression general treatments to single patient treatment and the balance to be done between especially possible effects of the treatment in terms of benefit and but also in terms of complications that with the introduction of all these new drugs, no one still of us knows about secondary effects of these patients and their role in the long term. I look if we have questions from the audience, but I do not see them on my screen. Do we have a question? Yes, Nada, please.
Very nice talk, Frederick. Like really, I'm so much in favor of doing biopsies, but what I fear by saying that every time is that people that are not as it's easier and easier, but again, it's not devoid of side effects. So I would really push and advocate that this is done when you have a clinical trial that you could offer to patients, and in a way that would also speed clinical trials that are really more comprehensive aimed to treating those DIPGs. I don't know what's your thought about that. I agree it's way more safer. But in any shape and form, if nobody's going to be acting more on that, that was my comment. And I had a question regarding Foundation One. What about if Foundation One doesn't give you any hit? Would you have other alternatives that you could use?
We struggle there, as does everyone else. If you don't have a molecular target that's targetable, then we really fall back on our standard conventional treatments, which is standard chemotherapy and radiation therapy. And we all know that, at least for the H3K tumors, that doesn't seem to be effective at all.
Absolutely.
Regarding your question, I think you're right. We can't get IRB approval to biopsy children just for academic interest. The only way we can get IRB approval to biopsy these children is if we have a protocol where it changes their treatment based upon what we see in the biopsy.
That's exactly right. Thank you. And if you need anything from, there are a lot of things that additional sequencing could give you sometimes for some tumors.
So we have a couple of questions from the audience. What about getting CSF and biopsy at the same time? This is both for you and Nada, I think. What's the suggestion about this? We may find differences, I guess the question is.
I think the future of liquid biopsy is bright. Right now, it's just investigational, but in certain centers, our group has a paper coming out on being able to profile medulloblastomas based upon CSF proteomics. I think in the future, we'll see that it may save us having to do brainstem biopsies in these children. At the moment, we're not there.
Okay, thank you. Do we have other comments from the people in the presenters audience?
So regarding CSF, I would add that it should be part of every clinical trial from now on. That's target midline glioma or any brain tumor. I think CSF is a mine. It's understanding what it gives us, and it's not only one point, but medullo is actually the easiest because it's part of standard of care to do lumbar puncture to assess if there is any. This is why it's easy there, but I think for every single brain tumor, if it's safe, and oftentimes it is, we should have CSF embedded as part of the practice going forward.
So agreement on taking a CSF sample in every patient, even
As
with
part
the
of a
IPG,
clinical,
not an
yeah,
agreement
but as part of a clinical
trial.
protocol, as part of a
Okay.
trial. Never do it on your own, and it's not safe
Okay.
for patients.
Okay. Thank you. Thank you, Nada. Thank